Okay, what I would like to tell you is now something really more in basic research. And first, I would like to present my laboratory to you. And this is located in Heidelberg. Very nice city here, with some very nice old buildings, and the the new university pediatric clinic. I take it like that. Okay, thank you very much. This is the new university pediatric clinic, which was just built about 2 years ago. We are engaged in the group of the GlycoKids, parents organization from Germany, and perhaps would be interesting for some parents to just overtake this it's not an email size, it's a web address, exactly. Computers are always a problem for me. Therefore, I've got my stuff there. And as you all know, glycosylation defects affect a lot, are multisystemic defects, which affect a lot of organs, and especially the parents will know that. And this is induced by defects in glycoproteins, and in underglycosylation of these glycoproteins leads to changes in the folding of the proteins, in the directed transport, biological activity, cell-cell interactions, and differentiation. And I will show you some pictures of mice. We are working mostly on mouse models. And it is the biochemical way is quite complicated, but in the last years, whole. Wo muss ich denn jetzt drauf? Ja. No, das ist etwas tückisch. A whole variety of diseases have been found in this way of glycoprotein production, and they affect enzymes in the cytosol, in the endoplasmic reticulum, in the Golgi apparatus, and this is quite complex in the meantime to investigate it in laboratory research. In our laboratories, or in most laboratories, the first routine diagnostics starts with investigations on isoelectric focusing and in changed isoelectric focusing patterns. I don't want to go too much into detail, but these investigations ends up in the identification of different types of CDG. And I would just like to give you just a short overview of what's happening in the laboratory. What we see by investigations of isoelectric focusing are changes in the glycosylation of normally completed of normal N-glycans linked to transferrin, and that are missing, like in early defects for CDG1, and shortened in defects of CDG2, mostly. Since there are some specialists sitting here in the field, like Hud and Thorsten, I just show you some of my co-workers. There's a sample preparation, and we can take samples from a whole variety of like urine and blood and cells, leukocytes, erythrocytes. There is then a scientific evaluation of the isoelectric focusing. My old campaign for nearly 15 years now, Christian Thiel. This is then afterwards a medical validation, and then it starts first with an investigation upon CDG-1A and 1B, and and this is the analysis of such investigations. But as I showed you, there are a lot of difficult CDG types besides of this area. We then start with labeling of fibroblasts, skin fibroblasts of the patients with radioactive manos, and perform high-performance liquid chromatography on them, preparation of the samples, scintillation detection, evaluation of the scintillation, and then afterwards the genetic analysis on patient cells, and for sure the data evaluation. So you see, this is just an example for a case of a disease we found in the H11 protein, and this is the DNA of it. You can see there is a change in the patient, and the parents are heterozygous for this mutation. In the meantime, we are also using whole exome sequencing to, especially to investigate unknown CDG cases, and so this is a whole machinery that we are working on. So let me now come to the topic of my talk, what are animal models. And I will just give a short introduction in generating animal models, and then show you and that there are a whole subset of animals which are used for these models. It starts with worms like Xenorabites, flies, zebrafishes, frogs, and mice. And most of our work in the last 15 years was bound to mice, and we are just starting, and this is not published, so this is pretty interesting. We just started with a frog model for CDG-1A. In the meantime, for a variety of different CDG types, mouse models were made, and I will talk about a mouse model for a CDG-1A, but I will start with the mouse model for CDG-2C, which is pretty interesting because we are just working on this on several organs where we found defects, severe defects. CDG-1C, also called pharmacological LAD-12, is a defect in the transporter of the Golgi transporter of GDP fucose, and it was Thorsten Marquardt, who gave his talk already, who found it first. And what is affected in these patients is this transporter, which brings fucose, the GDP fucose, into the Golgi apparatus, where the fucose is then translocated to newly synthesized glycoproteins. And exactly this defect of this patient, and it is shown here for the sugar residues. Oh, sorry, ich hab's falsch gemacht. And the fucose is this red triangle, and it is the fucose residues, or these fucose residues are linked to oligosaccharide in different positions. And as we found in our lab was that we can completely found a complete loss of fucose binding in patient cells. And by this way, and perhaps to give you an overview of what happens there, these are leukocytes rolling, and you should think about your own blood. These leukocytes are rolling upon your endothelium in your cells, in your endothelium, and in patients. They are very important. This rolling is very important for adhesion, transmigration, and a reaction against inflammation. And in case of patients, this doesn't happen anymore. So these patients are very severely affected by diseases. And it was the group of Thorsten Marquardt and Dietmar Festweber who found that treatment of the patients with fucose normalized these infections and the severe diarrhea the patients were suffering from. So this is just summary one. So fucose mediates many and various recognition, binding, and interaction processes, and the loss of the leukocyte rolling due to hypercoagulation leads to broad inflammation processes, and they are really life-threatening. Perhaps I should go on with it. And so this is the mouse model that we made in our lab, and astonishingly, this is really comparable to the human model. We have a severely reduced GDP fucose transporter activity, and we have a normal, or no embryonic lethality, let's say, but we have a high postnatal lethality, as you can say here. We have this leukocytosis and the seizures in the beginning, and so this is a very nice mouse model to investigate the patient's deficiencies. And we found defects in the lymph node with hypocellularity and primary follicle rudimentary or missing. We have severe changes in the lung, which we are investigating in the moment for proteins that are affected, fucosylated proteins that are affected in this area. And for the lung development, there's a very critical point in building normal lung tissue, and this is directly after birth, about day 2.5 to 5, and this is what we are investigating currently for fibroblasts, for fucosylated fibroblasts. And what we think is, that was already published by Bostrom, that FGF or PDGF might be involved in these developmental steps, because they showed already such difference as we see it here, and it is comparable. We are just looking upon that. The other thing, which is also not published, is that we found morphology changes in the brain. Although these mice are smaller, the hippocampus did not show any differences besides a smaller gyrus dentatus. And this is an area which affects adult neurogenesis. And in behavior studies, we found that there are changes, severe changes, and in fear conditioning of these mice. So, in fear conditioning of these mice, and we try to investigate enzymes, proteins, fucosylated proteins from this area of the brain in the moment. to find a mechanistical background on these behavior changes. And what we find so far in gene expression studies of the hippocampus, we found a variety of changed gene expression in cell adhesion, neurogenesis, exogenesis, synaptic transmission, protein transport, and so on. And in fucosylated hippocampus proteins, we found a variety of proteins which are involved in biological processes, and and they have special molecular functions which affect the catalytic activity, signal transduction, metabolic processes, protein binding, and for me, signal transduction is very interesting, and this is what we are investigating. So the SLC, also the CDG-2C mouse model, to say it in the old words, resembles in several aspects to the human disease, and in questions of this is one now. Growth retardation, leukocytosis, general hypofocusation, and it is possible to refucosylate the glycoconjugates by fucose supplementation. In the next model, we investigated defects in CDG-1A, so the most the common form of CDG, now called PMM2-CDG, which is located here, and this is more biochemical. The defect is in the conversion of mannose 6-phosphate to mannose 1-phosphate, and these are the most common features of the disease that most of you will know. It is also this brain which is affected, and you have these inverted nipples, scoliosis, as you can see here, and this classical picture of CDG-1A with the loss, or the partial loss, of oligosaccharide residues. In a knockout mouse model where we completely drop down the activity of the enzyme, we found that it is embryonally lethal at around the 8-cell stage, so very early lethal. And therefore, we decided to generate a hypomorphic mouse model for PMM2-CDG by bringing in genes or mutations which are comparable to human mutations, so that we had a residual activity as it has been observed in all patients so far. What we saw then, and this was very disappointing in the first time, was that at day 9.5 there are littermates, healthy littermates, and littermates with a mutation, and they started to become smaller. And after that, and you have to. This is a picture how big the size of these embryos are. So you have to deal with these very little embryos for cutting them in pieces and to investigate. And what was happening there is that these are littermates. So these are healthy brothers or what else, and from the same mother, and they went down and were resorbed, completely resorbed at day 12.5. So this was really disappointing because it took us a long time to prepare this, or to generate these mice. And it was our idea, coming out from research from Hutt's lab and our lab, that on fibroblasts, that you could perhaps elevate the level of mannose in the serum of fibroblasts, in the serum of the mice, by feeding the mice with serum before breeding them, so that the mother animals have already an elevated level of mannose, and the effect was pretty striking. At day 9.5 to 10.5, this is a very crucial date in embryonic development, and in many other animal models, this is the striking point where the embryos die, and what's happening there is that the heart and vascular system development is taking place, and that the neurologic development is taking place, and was, to my opinion, is very crucial for our embryos, is that differentiation of maternal and fetal blood circulation systems are coming together, and that this is not working, because as you will see now, here you can see embryos from the same age. They are much smaller, undersized, and they are already degrading. And this is, for example, the heart of a healthy litter mate, and the heart is already going down and breaking on. But what I think is crucial is you see the area of the placenta with severe bleedings in this case, and this is in comparison to the litter mate trophoblast area. Okay, this is a bit complicated. I'll go through it. Interesting is that when we fed the mothers with mannose-enriched water, we found, and if you remember, there is we have these smaller animals, and that they have been that they are completely destroyed at day 12.5. Now you see the mannose treatment on these mice, and they are comparable. And what's astonishing is that they are born and they are completely behaving completely normal. And this is the point where we are working on in the moment, on the proteins which are affected, and why this mannose treatment is so important at this time point. We don't know so far, but we are currently investigating it. So you can see for the organs of these mice at the age of 3 months, there are no difference visible in brain, heart, liver, and lung, thymus, kidneys. This looks all normal. I discussed this before, that there is a critical time point for the organ development, and it would be interesting to really, or what we are currently investigating are proteins, glycoproteins, which are changed at this critical time point. What we can show so far is that a hypomorphic mouse model for CDG-llc shows severe degradation and hyperglycosylation at embryonic tissues around day 10. and that the lethality can be prevented by supplementation of female mice with mannose, and which elevates the blood glucose mannose level. And come to my last point. It's also something really new and a bit crazy perhaps, but we started with a frog model for CDG-1A, and this was not a joke, but the frog is a model which allows us to have a high amount of embryos. They are easy to get, and this is an interesting story. It's one of my PhD students, and she showed it to me. I never did it by myself. You take the frog. These are quite big frogs. They are like that. They are from the size like that. Take it and just squeeze them. And then the eggs are coming out of the frog. And then you you just have the eggs, but you need to breed them, and this is really a bit brutal. You're taking a gonad you take a gonad of an egg of a frog, so not a human, a male frog. And you hack them into pieces, and you just go and give these pieces to the eggs. And in between of, let's say, 5 minutes, the eggs are turning to one side, and you see that there's a little black point appearing, and then you see, okay, these eggs are right, you can use them for further experiments. It was a very interesting method in former times. This is the cycle of development from the Xenopus, and I will show you some a picture in a minute about this stage at around day 4. But it is an interesting mechanism. Huh? It was called the pharmacist's frog, and this was used to see whether a woman is pregnant in former times. They just gave urine from women to the eggs of the frog, and when they showed what I described before, it was a sign that the women are pregnant, and therefore they are called pharmacist's frog. But it's Xenopus levis, a pretty big frog, and it's easy to work with these embryos because they are big and easily to manipulate. So what we did is, or what our idea is to investigate the signal transduction pathways which are affected in CDG-1A, and since these embryos are outside of the frogs, we don't have the problems that we have with mice where the embryos are inside. So it's easier to investigate the embryos, and this is good for signal transduction processes and patterning, which might be affected also in human beings. And so far we could show that with a so-called antisense morpholinear technique, which just drop down the activity of PMM2, so the enzyme which is affected in CDG-1A, we can drop this down to about 30% residual activity. And what we can show so far is that this reduction leads to leads to morphogenic defects in the backbone and in the and to eye dysmorphisms, and we are currently investigating the signal transduction ways which are involved in these or lead to these deficiencies. And we have so far reduced in the frog a reduced PMM2 activity, a general hyperfocusylation, morphological phenotype, loss of function due to hypoglycosylation, and that can be, and this is astonishing, and it seems or shows that this is really specific, that the injection of PMM2 leads to the rescue of this, these effects. So I will just would just like to show my group and thank all co-workers in my group, and thank you for your attention.